Hello everyone and welcome to another MCAT ACE lecture. In today's lecture we are going to discuss how genetic information can flow from genes all the way to proteins. We are also going to discuss the nucleic acid structure, how DNA replication happens, go over the enzymes that are required for DNA replication, what is the process of DNA proofreading, and also other mechanisms of DNA repair that prevents accumulation of mistakes in a DNA. Discuss codons and the genetic code, discuss different forms of DNA mutation, processes of transcription, post-transcriptional modification, as well as translation, then discuss chromosomes organization in terms of the heterochromatin and euchromatin, as well as centromere and telomere structure. And then finally we will have control of gene expression in eukaryotes compared to prokaryotes. And then as always we will conclude our lecture by going over some practice questions. Alright, so let's start right here with the nucleic acid structure and function. So basically the two types of nucleic acid that we are having is DNA or RNA. And DNA stands for deoxyribo, so deoxyribonucleic acid, while RNA stands for ribonucleic acid. And these are made of the linear polymers of nucleotides that are joined together by phosphodiester bonds. So phosphodiester bonds. Now, nucleotides are basically the building blocks of every organism's genetic material, and they are made of three components. Number one, they're made of a pentose, which is a 5-carbon sugar. They are made of nitrogenous bases, which include guanine, cytosine, adenine, and thymine. In RNA we have uracil instead of thymine. And then the third structure is the phosphate group, which has a negative charge, as a consequence of which DNA and RNA have a negative charge. Now let's talk a little more about nitrogenous bases. So basically, based on their structures, bases are divided into two groups. Purines, which are made of a double ring, example of which is shown right here, guanine, you see how it has double ring, and adenine. So these are examples of purines. And then we also have pyrimidines, which are made of a single ring, example of which includes thymine, uracil, and cytosine. So the memory aid that I'm using to memorize all these names as well as their binding, because guanine always binds with cytosine while adenine generally binds to thymine. I mean, there are exceptions when there's an error and they by mistake they bind to another base that they're not supposed to, but the correct binding is guanine binding to cytosine and adenine binding to either thymine in DNA or uracil in RNA. And the memory aid that I'm using here is called GASET. Alright, so with this memory aid I always remember that the first two are purines. So you see how we have purines for adenine and guanine, and these are double rings, while the second two are pyrimidines. And then in terms of binding, I always know that G binds C and A binds T. Or so adenine binds thymine in DNA, or adenine binds uracil in RNA. So that's about it. We've covered most of the high yield points you will need to know for the bases. Now, one other important point is that guanine and cytosine form 3 hydrogen bonds between themselves, while adenine and thymine, or adenine and uracil, are having 2 hydrogen bonds. And the way I usually memorize it is that G and C, they're so big that they can hold 3 bonds, while A and T, you see, we can't really put 3 bonds in here. So G and C, it has it looks to me having higher surface area that we can put in 3 bonds. So that's how I memorize it for myself. If you have any other means of memorizing all these concepts, then by all means, you don't have to use any of this. It's just to help you with memorization in case you have a problem. All right, now that we've discussed all these points, one other important point that I'd like to mention is that if we are having 2 DNA strands, let's say we have DNA number 1 that is 90% made of guanine and cytosine, and then we have DNA number 2 that is 90% made of adenine and thymine binding. So this one has adenine and thymine, while the other one had guanine and cytosine. What would be the difference in terms of the strength of binding of these 2 double strands? So DNA number 1 and number 2, which one is bound stronger? And the answer is number 1, because guanine and cytosine has 3 hydrogen bonds versus adenine and thymine, which have only 2 hydrogen bonds. So if we have a DNA strand that has a high GC content, then that DNA strand is more tightly bound, and usually we need higher temperature in order to denature that DNA strand. All right, so that's one important concept that examiners like to test on MCAT. Now that we've reviewed all this, I'd like to go over one important difference between RNA and DNA. So DNA is made of a deoxyribose sugar, which means that its second hydrogen on its second carbon, it has a hydrogen group. It doesn't have oxygen. That's why it's called deoxyribose. So DNA has deoxyribose sugar versus RNA that has a ribose sugar, where on its sugar second carbon there is an OH group. So that's one important difference between DNA and RNA. The other difference, as we just discussed, in terms of bases, DNA uses G, A, C, and T, guess it, while RNA uses G, A, C, and U. So it's A and U that bind together, C and G, and in DNA it's same thing, G and C and A and T. And then finally, one other important difference is that generally speaking, DNA is most of the time double-stranded. So this is double-stranded, while RNA is usually single-stranded. Of course, there are examples of viruses that are made of a double-stranded RNA or single-stranded DNA, but I'm just saying, generally speaking, DNA molecules are usually double-stranded versus RNA that is single-stranded. Now, an example of DNA that is double-stranded is humans, for instance. And an example of RNA is HIV virus, where its genome is made of a single-stranded RNA. Now, because of the fact that DNA is double-stranded in humans, that's why that their structure is very stable. It's very hard to have mutations in our genome. Versus HIV is made of single-stranded RNA. And so it's very easy to have mutations in HIV virus. And that's one important reason that the HIV virus can easily develop resistance to different drugs. Because of the high rate of mutations, the structure of HIV continues to alter, and so it will evade different medications that we are using. Now, going back to the DNA and its structure. So the first scientists who have proposed the helical structure for DNA, that they proposed that it's composed of the 2 helical strands, were Watson and Crick. And how did they come up with that conclusion? So basically they used 2 different findings to come up with the conclusion that DNA is made of 2 helical strands. One, they used the Chargaff's rule. And according to this rule, the total amount of purines in a DNA molecule is always equal to pyrimidines. And their conclusion was that regardless of from which species we extract DNA, always the number of purines to pyrimidine is 1 to 1. And if you think of it, we have gaset, right? So one purine always binds to one pyrimidine, and then the other purine always binds to the other pyrimidine. So that's why the ratio is 1 to 1. And then the other finding that helped Watson and Crick come to their conclusion was that they used the Franklin's X-ray photographs of DNA, which has shown a crossing pattern of dark spots that has suggested that DNA is probably having a helical structure. So based on these findings, in 1953, Watson and Crick published their description of DNA. They hypothesized that it's two helical strands of DNA that are wound together, and then they suggested that these DNA strands during replication probably will unwind, separate from each other, and form a new one. And it was amazing how in subsequent years, everything that Watson and Crick have predicted actually came true. Now we know that DNA is made of a helical strand, and that it replicates via the semi-conservative mechanism where only one strand of the parents remain in the replicated strand. So here, if you have two original strands, after these two separate from each other, and then a new DNA strand will be formed. So it's semi-conservative because only half of the parental DNA has been remained. Now, I've earlier discussed the structure of DNA, so here now I have an image with you so that I can go over that again. So a combination of the phosphate group and a pentose sugar in a DNA is referred to as backbone. Same with the RNA. So a combination of pentose sugar and phosphate, these are all DNA backbones, and these are being held together by phosphodiester bonds. So here you can see that the pentose sugar is being connected to the next one via a phosphodiester bond, and same here. So the order of binding according to the site that the phosphodiester is bound gives us the direction of the DNA strand. So here we can see that we have the 5' carbon and then the 3' here. So the direction of DNA in this example is 5 to 3'. And so here we have 5 to 3' DNA strand versus 5' to 3' DNA strand. And then another important point is that, as we just discussed, phosphate group has a negative charge, as a consequence of which DNA is also negatively charged, and thus it's hydrophilic. So DNA has a negative charge and it's hydrophilic. And then the final important point I would like to mention is that it's the nitrogenous bases right here. Let me pick a different color. There we go. It's the nitrogenous bases that determines our genetic code. So in this DNA example, for instance, you can see our genetic code here is determined by all these nitrogenous bases that are shown right here. Okay, and the two strands are held together by the hydrogen bonds. Okay, now moving on to the mechanism of DNA replication and which enzymes are required. So basically, if you recall from our meiosis and mitosis lecture, the cycle of cell division is divided into G1, S, G2 phase, and then finally mitosis and cytokinesis. So G1 and G2 are the cycles during which the cell will grow. And then S phase is when DNA replication actually happens. And then during mitosis, that's the time that chromosomes will separate into separate poles, and then finally during cytokinesis, the cytoplasm will also divide. All right, so here we would like to discuss the mechanism of DNA replication and the enzymes that are involved. So first thing that I would like to mention is the difference between prokaryotes and eukaryotes. Prokaryotes, like for instance bacteria, they're having circular DNA. All right, so they will start their replication only at one point. They have a single origin of replication, and they will start to spread out from that point and give rise to two circular DNA. Eukaryotes, on the other hand, they have a straight DNA. It doesn't come as a loop to each other. It's just a straight DNA. And so therefore they have multiple origins of replication, because if they wanted to start on one point and wait until it goes all the way to the end, it would take forever. So therefore eukaryotes have multiple origins of replication, from which points the DNA will unwind and then replication enzymes will start to replicate. So how does replication actually happen? So replication takes place at the replication fork. And so here we have an example of replication fork here, another replication fork here. So the point where the DNA has been unwinded is referred to as replication fork. And this unwinding is done by the helicase enzyme. So you can see how here the DNA is opening up. That's the work of helicase. And so is with the other DNAs. All right, and here I have a drawing for you that summarizes the function of every one of these enzymes. So we have the helicase that will unwind the DNA at the replication fork. But now what happens as we are unwinding DNA? Since this is a helical structure, there would be supercoiling that will happen at the end of the DNA strand. So imagine if you're having one of those old phones that has a cord that is twisted on itself. If you try to open the cord, you will see that the rest of the cord will become super coiled on top of each other, and we don't want that to happen. So here I have an image that I've got from Wikipedia. It shows you how in prokaryotes we have a circular DNA, and as we are trying to open it up, it will supercoil on itself. And if this happens, then we can't continue the DNA replication. Now, in order to avoid this problem to happen, we have the enzyme called topoisomerase, which nicks the DNA in order to relieve all the supercoils that is being generated by unwinding of the DNA. So that's the job of the topoisomerase. Now, as DNA is being opened up, now we are ready to start synthesis of a new strand, and that is being done with the help of 3 different enzymes that are listed here. So the first enzyme that we need is an RNA primase. Basically, RNA primase will synthesize a small RNA primer that acts as a template, and once this is formed, then RNA polymerase can bind and continue to form a new strand of DNA. So basically what DNA polymerase 3 does is that it will line up along the 2' strand and establish the complementary pairing. So if there is an A on the 3', then it will add a T on the 5'. If there is a C, it would add a G. So a complementary pairing will be initiated by the DNA polymerase enzyme. But this enzyme can only synthesize can only add nucleotides on the 3' end of the growing strand. So here you can see that always the growth of DNA is from 5' to 3' direction. As mentioned here, growth is always 5' to 3' direction. Now with this limitation, there would be a problem, because as we are extending the DNA, as we are unwinding it in this direction, the leading strand right here, which is continuous, will not have any problem because it can continue adding on and on and on. But then the problem is that we have another strand that is interrupted, and it's called lagging strand, because as we are opening it up, this region remains empty. We can't really add nucleotides at the 5' end. Only nucleotide can be added on the 3' end. All right, and so that's why this is called a lagging strand, because it's interrupted. And these fragments are referred to as Okazaki fragments. So what happens here, as we are opening up DNA, new RNA primase comes in and open a new template, and then again polymerase comes in and form a new fragment. As we open up more DNA, another fragment will be made, and so on. So the lagging strand is made of numerous fragments. Now once these fragments are made, they will be eventually joined together with the help of the ligase enzyme. All right, so ligase enzyme is used to join the DNA fragments that are in the lagging strand. The other name for them is Okazaki fragments. All right, with that overview, Now we can summarize the findings in this table. So the type of enzymes that we are having here, we have the DNA polymerase 3, which synthesizes DNA in the 5' to 3' direction. It also has an inherent 3' to 5' exonuclease activity for proofreading. So what it means is that as new nucleotides are being added right here, so here we are having new nucleotide that is added. If this nucleotide is added by mistake, the DNA polymerase 3 can remove it by its inherent exonuclease activity. So it can go back, take it off, and put the correct one here. So let me give you an example. Let's say we had A here, and mistakenly DNA polymerase 3 has incorporated G into the growing strand. That's a mistake. So by proofreading mechanism, DNA polymerase will remove the G and replace it with a T. And this proofreading ability of DNA polymerase will significantly reduce rates of mistakes during DNA replication. So once again, synthesizes DNA in the 5' to 3' direction. It also has an inherent 3' to 5' exonuclease activity. So if there is a mistake, it's on the 3' end that they made a mistake. So it can go backward from 3' to 5' direction. It can have exonuclease activity in order to remove this mistake and replace it with a correct nucleotide. Now with DNA proofreading, misincorporated nucleotides can be removed immediately. Then we have the RNA polymerase 1, which will degrade the RNA primer via its 5' to 3' exonuclease activity and replace it with DNA. So you know how RNA primase was used to create all these templates. In the end, DNA polymerase 1 will come in and replace all these RNA templates with an actual DNA. As we just discussed, we have the primase, which creates a template of RNA on which DNA polymerase 3 can initiate the DNA replication. DNA ligase is used to join the Okazaki fragments via the formation of the phosphodiester bonds. Helicase was used to unwind the DNA at the replication form, but this process creates supercoils, and these supercoils are relieved by topoisomerase enzyme, the other name for which is gyrase. So sometimes students ask, so what's the difference between topoisomerase and gyrase? So let me mention that there are different forms of topoisomerase. There is type 1, which creates only a single strand cut in DNA. And then we have type 2, which creates a double strand breaks. Now, gyrase is a type 2 topoisomerase that is found in bacteria. But in any case, for MCAT, what you need to know is that topoisomerase or gyrase are the enzymes that are involved in relieving the supercoils by creating nicks in DNA helix. All right, now moving on to the next concept, what happens after DNA replication happens. So after DNA synthesis, each DNA copy will now have a new strand and an old strand. So here you can see that the parental strand, they were separated from each other, shown in green, while a new strand in red is created. So we are only having half new and half old. That's why this is referred to as semi-conservative replication. Now, in bacteria, there won't be any problem or shortening of DNA because their DNA is circular. In eukaryotic cells, however, there is a problem because with each replication, as the RNA fragment is removed from the new strand, the 5' end will shorten, and the amount of shortening is equal to the size of a primer, the RNA primer that was used to fill that space. So the telomeres of chromosomes with every replication becomes shorter and shorter until they reach a point that they can no longer divide, and that's actually called cellular aging because every cell has a finite number of divisions. They can divide, for instance, 40 times, after which they die. They can't do that. So they're limited in their number of divisions. However, there are exceptions because we are having actually some immortal eukaryotic cells that can divide indefinitely, and these examples of immortal cells include, for instance, cancer cells, germline cells, and stem cells. But how do these cells divide indefinitely? Aren't their telomeres keep getting shorter and shorter? And the answer is that these cells have a new enzyme called telomerase. And the function of telomerase is that after every division, it just adds an extra sequence to the end of DNA. So therefore, with every replication, those extra sequences that are made by telomerase will be removed. The actual genetic coding of the cell will not be affected. All right, now let's discuss the telomerase and how it works. So here I have an image of the telomerase enzyme for you, and you can see that it's made of a reverse transcriptase enzyme. And basically what reverse transcriptase does is that it converts the RNA into DNA. So the telomerase has an RNA template right here, and with its reverse transcriptase enzyme, it will create a new DNA strand at the end of telomere. All right, so telomerase is made of an RNA template and a reverse transcriptase enzyme which converts RNA into DNA. And by doing this, they would add extra strand at the end of the DNA to prevent loss of DNA from every replication. All right, now before I move on to the next concept, reverse transcriptase enzyme. That's a hot topic that is being frequently asked on MCAT. So what I would like you to know is that HIV is also having RNA genome, and how it will replicate is by conversion of its RNA into DNA, and that is also being done by the reverse transcriptase enzyme. Okay, so just one more time to clarify, I don't want any confusion. So we know how DNA via the transcription process is being converted into RNA, and now RNA via the translation process is being converted into protein. Now, if you're having HIV virus, via the reverse transcriptase enzyme, it will first make DNA and then RNA and protein. Now let's move on to the next concept, which is DNA proofreading and DNA repair. Alright, so we've already discussed this first concept, which is DNA proofreading. We discussed how during DNA replication there could be error in incorporating a wrong base. And the rate of error is about 1 error in every 10 to the power 4 to 10 to the power 6 base insertions. On the other hand, DNA polymerase 3 has inherent proofreading ability by having the exonuclease enzyme. So the 3' to 5' exonuclease activity of DNA polymerase 3 allows this enzyme to remove any misinserted bases. I'm not saying that it's going to be perfect without any error. There could be still errors, but the number of errors significantly drop with this property of DNA polymerase 3. And actually, from 1 mistake in every 10 to the power 4 to 10 to the power 6, it drops down to 1 in every 10 to the power 10 base insertions. So this DNA proofreading ability of DNA polymerase 3 is a very important mechanism in preventing errors during DNA replication. Now, what happens if there is error? The error will happen. There will be misinsertion of bases, or later on there could be damage to DNA. So the question is, how can we now repair the damaged DNA? So there are multiple mechanisms through which DNA can be repaired, and these mechanisms include base excision repair, nucleotide excision repair, mismatch repair, as well as repair of a double-stranded break. So let's go through every one of these mechanisms. The very first one is base excision repair, which is I would say the most simple of all, because it would actually repair DNA without distorting its structure of DNA helix. So what happens is that by using the glycosylase enzyme, throughout the base excision repair, the damaged base will be removed. So what glycosylase enzyme does is that it will cleave the N-glycosidic bond that connects base to its deoxyribose sugar. So now the base has been removed, and all we are having here is a backbone. So the other name for this structure is A basic DNA. And what A basic DNA means is that a DNA that doesn't have a base. Next, there would be endonuclease enzyme that will hydrolyze the phosphodiester bonds and remove now the A basic DNA. And then finally, DNA polymerase and the DNA ligase will come in to fill in the gap. So that was a simple mechanism that was induced by the base excision repair. The next, a little more complicated mechanism is nucleotide excision repair, which uses as many as 30 different proteins in order to carry out this process. So what happens with the UV radiation is that it will cause formation of the pyrimidine dimers. So pyrimidine dimers are formed from exposure to UV light. Now, the job of the nucleotide excision repair is to remove these pyrimidine dimers. And how it's being done is that first with the endonuclease enzyme, it will recognize the lesion, nick the surrounding DNA, and then with the help of the helicase, unwinds this section of DNA and remove it. And then finally, DNA polymerase and DNA ligase come in to fill in the defect. The next mechanism is mismatch repair. So you know how we said we have proofreading, but it's possible that proofreading makes a mistake. And so by the time we are done with our DNA replication, now we need the mismatch repair mechanism to kick in and repair for any mistake that is there. So let me give you another example so that you know the difference between proofreading and mismatch repair. All right, so as I'm here writing for you, I would say I'm writing a letter, and all of a sudden I notice I misspelled letter. It should be L-E-T-T-E-R. So what I did right here was proofreading. Instead of saying letter, I went back and changed the last word. So that was an example of proofreading. And now I'm done with my sentence. Now, what mismatch repair does is that I go back and reread my sentence, and I'm like, oh, I've got another mistake right here. I should have said I'm writing a letter. So that's an example of mismatch repair. Proofreading repairs only the last word, versus mismatch will go and check the entire sentence to see what mistake we have there. All right, so here is an example of mismatch repair. DNA replication has progress, but now we do have an error there. Now the question is, once the mismatch repair enzymes go in, which side of the DNA should they repair? Is it this one that is an error, or is it the bottom one? Because let's say here we are having A that has paired with G. All right, we know that this is a mismatch, but which one is a mistake? So what happens is that the parental strand has been methylated, right? So this way the mismatch repair enzymes know that this strand they shouldn't touch. It's the parental strand. So that's how they know that now they should repair the other strand. So the mismatch repair would recognize the lesion, then with the help of exonuclease and helicase enzyme, it would cut and remove, and then DNA polymerase and ligase, just like the other cases, will kick in and fill the gap. All right, and then the last mechanism of DNA repair is repair of double-stranded breaks. So let's say that from a radiation we have a double-strand break. Now how are we gonna repair that break right there? So there are two different mechanisms through which double-stranded breaks can be repaired. One is called homologous recombination. So homologous recombination. and the other one is called non-homologous end joining. So what happens with the homologous recombination is that this is basically an error-free mechanism, and what it does is that it uses the information from the sister chromatid in order to repair it. So you know how every chromosome is made of two chromatids, so we have a sister chromatid right there. So instead of trying to find out what has gone wrong there, it will go to the blueprint of what is there. It will find the other chromatids and use the information there in order to repair the break. And this is called homologous recombination. On the other hand, we can have non-homologous end joining, which can cause errors because it doesn't care here what we've lost here. What this mechanism basically does is that it just simply joins the two ends of broken DNA to each other. And so homologous recombination is restricted only to the S and G2 phase of the cell cycle, while non-homologous end joining can be done in any phases of the cell cycle. And so that's why in certain situations we have to rely on non-homologous end joining, because homologous recombination can only be done in certain phases. All right, now that we are done with the DNA replication, the next topic is genetic code. How does the information in our genes flow from DNA into proteins? And so basically the genome consists of the genetic material of an organism, which can be either in DNA or in RNA. An example of an RNA we just discussed is like an HIV virus, which via the reverse transcriptase enzyme produces DNA, and then that DNA will be transcribed to RNA and then translated to protein. And then we have organisms like humans that have DNA. Via transcription, they form RNA, and then via translation, they form protein. And so what I would like to do next is to go in more details regarding mechanism of every one of these processes. But before I do that, first I would like to talk about the genetic code. So if you've noticed, all the coding of the nucleotides, we refer to them as triplets. You see how I've highlighted triplets right here. And the reason for that is that our codons or our genetic codes are in triplets. And what it means is that every three nucleotides that are joined together, they would code for an amino acid. And I'm going to give you an example right here. So here you can see that AUG is coding for methionine, or like for instance GCC is coding for alanine, or GGG is coding for glycine. So the triplet codons are coding for different amino acids. All right, now the question that I have here is, we said that every codon is made of a triplet code. So the question is, how many different bases or nucleotides can we have in every one of these positions? Well, we already know we have G, A, C, and T, or uracil for RNA. So basically we can have 4 different bases for the first position. We can have 4 different combinations again for the second position, and then 4 different combinations in the third position. So the total number of triplets codes that we are having is 4 times 4 times 4 is equal 64. And here this table is showing you that we have 64 different triplets codes right here. But the total amino acids that we have in our body is only 20. So what happens if we have 64 codes but only 20 amino acids? Well, what it means is that some of the amino acids are being coded by more than one codon. And so here I give you an example. For instance, like leucine is coded by CUU, CUC, CUA, and CUG, as well as UUG and UUA, right? Proline is being coded by CCU, CCC, CCA, and CCG. Of course, there are examples where less codes are used, like for instance, UAU or UAC for tyrosine. So just keep that in mind. We have 64 different codons, but these are only coding for 20 amino acids. And that is the reason that some amino acids are coded by multiple codons, while some other ones are coded by as few as only one. All right, now the other important concept that I would like to show you here is the start and stop codon. The question is, you're having an mRNA. From where should you start synthesizing the protein, and from where should you end your synthesis? And so here you need a start codon to give you the signal that, oh, from here you should start your translation. And then there should be a stop codon that tells you, oh, at this point you're ready to stop the mRNA translation. And so the code for start codon is AUG, while the code for the stop codon is, here we are having it right here, UAA, UAG, and UGA. Okay, so in the example shown right here, you can see that we've started translation from this point that we had AUG, and as soon as we reach one stop codon, UGA, we have stopped the translation. And then the final question I would like to ask you is that what is a wobble hypothesis? All right, let me pick a different color in order to explain this concept for you. So you know how a codon is made of a triplicate code. So first nucleotide, second nucleotide, and then third nucleotide. What wobble hypothesis tells you is that base pairing with the third nucleotide is very relaxed. So there could be more than one complementary bases. In other words, base pairing with the third position of a codon could deviate from Watson and Crick rules due to the lower specificity. So here there is lower specificity, as a consequence of which not necessarily all the time Watson and Crick base pairs will take effect. Like for instance, A can go with G, or C can go with T. Let me write gas set. You know, C is supposed to go with G, so C here goes with T. So mispairing is more common in the third position compared to the first and second position. And so therefore, by having this property, cells are kind of relieved from the need to synthesize a tRNA for every codon. And if you pay attention, you can see that this is the third position that they don't care like how it changes. It still codes for the same amino acids. Or right here, the third position for leucine, doesn't matter what kind of pairing happens there. Even if there is a mutation or even if there is an error, it would still code for the same amino acid. So therefore, that's why the wobble lowers the significance of DNA mutations that happens on the third base of the triplicate codon. Now, talking of mutations, what type of mutations can we have? So there are different forms of mutation, which I've listed them right here. We have silent, missense, nonsense, and frameshift mutation. But before I explain that for you, let me show you. If we have a mutation in any one of these wobble positions, let's say for leucine, U, C, A, G, any one of these mutations, it doesn't really matter. We're still going to code for leucine. So this is an example of a silent mutation. All right. So silent mutation, mutations that code for the same amino acid, often associated with a base change in the third nucleotide position or wobble position. Now, how about missense mutation? Well, when we have a mutation that changed the amino acid is an example of missense mutation. And let me give you an example here. If here we have a mutation that changes A into G, then we are having alanine incorporated instead of threonine. And so that's an example of a missense mutation. Now, how about nonsense mutation? When a mutation converts an amino acid into a stop codon, that would be an example of a nonsense mutation. So let's say that here for tyrosine, C is mutated into A. We will have UAA, which is a stop codon. And this mutation is not very benign, and the reason for that is that it will truncate the size of a protein. So the memory aid that I have for you is stop then nonsense. So nonsense mutation promotes formation of stop codons. And if I go back to an example here, if you're having a mutation that, for instance, trying to look, we don't have a good example here, but let's say if we had a few mutations that would convert this one into a stop codon, then all we are having is a protein that has 2 amino acids. So nonsense mutations will cause truncation of the polypeptide that we are synthesizing. And then finally, we can have frame shift mutation, where if there is an addition or deletion of the nucleotide, there will be misreading of the entire downstream nucleotides. And let me show you what I mean by that. So let's say that here I either delete one of these nucleotides, or let's say that I add one. Let's say I delete one, that's easier. So let's say here I delete this amino acids. So after the start codon, what do we have? This would be the next codon, and then CCG would be the next codon, and then AUG would be the next codon. So if you refer to the table that I have right here, now we are having a completely different sets of amino acids that are being incorporated. And so therefore frame shift mutations usually can change the entire structure of the downstream proteins that are being translated. All right, so that covers discussion of the DNA mutations. Next, I'd like to go over the process of transcription. We already discussed what happens to DNA replication. We already discussed the genetic code and codons that help convert the mRNA into blocks of amino acids that builds the protein. Now I'd like to again go back and describe what's the purpose of having this transcription going on. So as you know, all the cells in our body have the same genetic content. I'm giving you an example. Our adipose tissues, our heart muscle, our nerve cells, our kidney cells, every one of them have the same DNA codes and the same genes, but their functions is totally different. There's a huge difference between our muscle cells and our nerve cells and so on. And how are they so different if they all have the same genes? And the answer is that every different cell expresses different genes. And so therefore transcription is a cellular means of regulating gene expression. And that's what leads to cellular differentiation between different cell types. So what happens with the DNA is that as the RNA polymerase transcribes RNA, here we are having an mRNA, which is a messenger RNA, and then throughout the translation process and with the help of tRNA, amino acids will be added together and form a polypeptide, which will later on fold on itself to carry out different biological activity. So you might have noticed that I've talked about mRNA, tRNA, and we also have another type of RNA called ribosomal RNA. So let's go over the difference between every one of them. So first we have mRNA, then we have rRNA, and then we have tRNA. The first one, mRNA or messenger RNA, it's basically the RNA that carries genetic information that are copied from DNA throughout the transcription process. And this mRNA carries codes as codons, which are 3 base sequences that are specific for different amino acids. So here I'm showing you an example of an mRNA that is carrying codes in codons, which are which are triple bases. Then we have the rRNA, which stands for ribosomal RNA, and this RNA is being produced in the nucleolus, so not nucleus, nucleolus, and make up the bulk of the ribosome. And then finally we have tRNA or transfer RNA, which carries amino acids to the ribosome for their assembly into proteins. And this tRNA basically has a clover leaf appearance, and there is amino acid at one end and an anticodon at the other end, and this anticodon will pair with the codons to determine, okay, for every one of these triplicate codes, what amino acid should incorporate once they are working with their ribosome. All right, so that's a quick overview of these processes, and I will go in a little more detail in a little bit, but first I would like to overview. So here again we have another image that overviews all these processes. So in the nucleus we have DNA that throughout the transcription process forms mRNA. Now the reason we call this pre-mRNA is that once an mRNA is formed, it still has to undergo certain modification in order to form a mature RNA. And this is called post-transcriptional processing, which involves, for instance, adding a 5' cap, adding 3' polyadenylation, and then finally splicing process. So there are different processes that needs to be done in order to season this pre-mRNA and form a mature mRNA. Now once mature mRNA is formed, it will go through the ribosomes, and then the tRNA will come in, match their anticodon with the codons of the mRNA, and ribosomes will help incorporate amino acids into a chain to form a growing polypeptide or protein that will carry out different functions. All right, so now that we have an overview of this entire process, let's go in a little more detail with every one of these steps. So the first step was transcription, which is being carried out by the RNA polymerase enzyme. So the RNA polymerase uses the template DNA strand, the other name for which is antisense, in order to synthesize the RNA. So here you can see that our template DNA in this case is the red strand, which is being transcribed. So here the RNA polymerase is actually moving from 3' to 5' direction. So that's the direction that the DNA is moving through. So it's like a zipper that's going through and opening it up, allowing for mRNA to get transcribed. And by going through in this direction from 3' to 5', so here we have the 3' side of DNA, and then this side is the 5'. So now it allows the mRNA to grow from 5' to 3' end. Because if you recall, DNA and RNA, they can only be added, they can only grow in the 5' to 3' direction. So that's the only side that new nucleotides can be added. We can't add nucleotides at the 5' end. So here we have 5' to 3', and so as new ribonucleotide enter, new ones can be added to this growing chain. All right, now here we have a simplified image that shows how template DNA strand, the other name for which is antisense, is used to create an mRNA. Now the other strand is called coding or sense DNA strand. And the reason it's called coding DNA strand is that the code for this DNA strand is identical to the RNA that is being transcribed. The only difference that you're having here is that since this is DNA, we have thymine, while this is RNA, we have uracil. But otherwise, ignoring this difference, their code is the same. So template DNA strand is used for transcribing mRNA. The mRNA that is transcribed, its sequence is similar to the coding DNA strand, with the difference that RNA has uracil versus DNA has thymine bases. All right, so now we have a pre-mRNA that is formed. The next step is post-transcriptional modification, and there are 3 different processes that happens here. The first one is addition of 7-methylguanosine cap at the 5' end. And then the second process is addition of poly-A-tail at the 3' end of the mRNA. And these processes help protect mRNA from exonuclease enzyme. So we've added a 5' cap and a 3' polyadenylation process in order to stabilize our mRNA molecule. Now there is a third process that can happen, and that is splicing. So in eukaryotic cells, the genetic code is interrupted by sequences that are actually non-coding DNA, and those sequences are called introns. So here in this example, exon is our coding DNA. This other exon is our coding DNA, but this intron is a non-coding DNA. It doesn't code for any proteins. So once this mRNA is formed, we need to cut this intron out. And the memory aid that I have for you is called introns out. So introns need to be cut out. All right, and the cutting of intron is done with the splicing process. The name of the enzyme is spliceosome that will cut the intron out, and so now we have a DNA that is an mRNA that is made of only exons, which are the coding sequences. But then we also have another process called alternative splicing, which selectively splice out certain exons from pre-mRNA. And the reason this process exists is that it gives a higher coding capacity for the genome. Because if you think of it, we have about 20,000 genes in our genome, right? So does it mean that we can only produce 20,000 proteins? If we didn't have alternative splicing, the answer would have been yes. But with the alternative splicing, it's as if you're shuffling cards. Imagine if you have a deck of cards and you keep shuffling them, how many different combinations can you have? So here, by having different pattern of splicing and leaving some of the so by different pattern of splicing, you can produce different number of proteins. And here in this example that I've taken from Wikipedia, I'm going to show you how it's being done. So in the first one, you're having exon 1, 2, 3, 4, and 5. So this is a full-length gene that has been translated. In the next case, we are having intron in between exon 1 and 2 removed, but then between exon 2 and 4, the entire sequence is removed. So by removing these introns, exon 3 is also removed. And so here we are having a truncated form of protein. And then in the third example, you can see that exon 4 is missing. So during the splicing of these introns, this entire sequence has been removed. So by doing this, we will get more variety of proteins from a single gene. And this is called alternative splicing, which provides an evolutionary advantage by expanding the coding capacity of our eukaryotic cells. All right, the next process is translation. So let me go back to the summary again. We've shown how transcription happened, how pre-mRNA has undergone 5' capping, polyadenylation, and splicing. Now we have the mature mRNA that is undergoing translation. All right, so we are down to translation. So translation is a process where the mRNA, where the information in the mRNA is translated into protein synthesis. And so therefore, amino acid sequences of the proteins, what you get right here, these amino acids, reflects the code that is encoded inside our mRNA, which was similar to our sense or coding DNA. So here we had the sense or coding DNA. The sequence of proteins right here that we are getting, the sequence of peptides, is a reflection of coding DNA. Now regarding the ribosomes, we just discussed that there are ribosomal RNA that is being assembled in the nucleolus. So basically inside the nucleolus there is RNA polymerase 1 that combines, that assembles the ribosomal RNA with the ribosomal subunits. And so here you can see that the eukaryotic ribosome is different than prokaryotic ribosome. We have the 60S and 40S subunits in eukaryotic. Once you combine them, it's called ATS ribosome. versus in prokaryotic we have 50S and 30S. Once you combine them, it's called 70S. I know their addition doesn't add up, but it's just a concept that you will have to know, that combination of 60S and 40S, we are referring to them as 80S ribosome in eukaryotes, versus prokaryotes have a 70S ribosomes. And that's actually one reason that the drugs that we are using to target bacteria are often targeting the bacterial ribosome, because their structure is different, and so we are targeting protein translation in bacteria without affecting our own cells, because the structure of ribosome in bacteria and humans is different. Now, once the mRNA is running through all these units, so the mRNA is now running through the ribosome, the codons that we are having here is being paired with the anticodon of the tRNA. As a consequence of which, the amino acids that is being carried by the tRNA is being added to the growing chain of a polypeptide. And here I have another image that shows you how ribosome will allow growing chain. So the transfer RNA passes through the ribosomes in the following order: A, P, and E. First it comes to the point A, then P, and then E. So the way you can memorize it is APE, like Planet movie Ape, right? So APE. A, P, and E is the direction that the tRNA incorporates. Now, once it enters at the A site, ribosomal A site, that's the site that it accepts the incoming tRNA. So here is the memory aid. A site accepts the incoming tRNA. And then we have the P site, which accommodates the growing peptide. So here the amino acid at the P site is being added to the growing chain. And then we have the E site, which, as the name implies, it stands for exit. So the tRNA exit the ribosome from the E site. All right, and then as the protein is being translated, the last step is post-translational modification. So now we have to undergo post-translational modification. And the changes that you see during this stage includes, for instance, covalent addition of a functional group, addition of a functional group, there could be proteolytic cleavage of the protein in order to convert it from inactive form to an active form, or there could be folding processes in order to fold the sequence of polypeptide into an enzyme or a protein that will carry out a specific function. So after the polypeptide is formed, it's not the end of the story. The protein still has to undergo either covalent addition or proteolytic cleavage or folding changes in order to form into a mature form of protein. All right, and that basically concludes the discussion of how we have gone all the way from DNA to synthesis of a protein, where through the post-translational modification it's now ready to undergo biological activity. Now let's go back. The next concept is chromosomal organization. So we know that the eukaryotic chromosome, it's quite large. It actually can be as large as 2 meters, our DNA. So how does that 2 meters of DNA is actually fitting inside a human cell, right? So it has to be packed and very organized in order for it to happen. So in order to achieve this goal, DNA is wrapped around a protein called histone. So here you can see that we have histones. So here is our DNA that is wrapped all around the histone. And the combination of a DNA that is wrapped around histone is called a nucleosome. So histone combined with the DNA wrapped around it is called a nucleosome. And then this combination of DNA and protein, which is called nucleosome, forms a string of beads which will organize into a highly supercoiled structure called chromatin. And then during cell division, chromatin actually reaches its maximum folding capacity and takes the shape of a visible chromosome. All right, so that's how we go all the way from DNA into visualizing chromosomes under microscope. Now, there are different terminologies I would like you to know, and those include, for instance, heterochromatin and euchromatin. So heterochromatin is a tightly packed DNA, and it's a repetitive form. So this is tightly packed, and generally it contains non-coding DNA. Of course, it has been shown in organisms like Drosophila that their heterochromatin can have a few genes, but generally speaking, compared to euchromatin that is rich with coding DNA, heterochromatin is mainly non-coding DNA, and it's tightly packed because it doesn't have any genes really to use up, so it's tightly packed. Euchromatin, on the other hand, is lightly packed. So let me pick a different color for here. So euchromatin is lightly packed form of DNA, and it contains coding DNA that can undergo transcription. So it's mostly our euchromatin that is being used for the transcription process. And then we also have centromere and telomere. So these two, centromere and telomeres, these are examples of heterochromatin that are tightly packed and don't really have any genes. So let's go over every one of them. The first one is a centromere. It's basically the indented area that is near the center of the chromosome, and it's used to link the sister chromatids together. So here we have the centromere right here that is holding the two sister chromatids together. Now I'd like to remind you the phases of mitosis. Of course, we have a separate lecture that we go through every one of those steps in detail, but if you recall, during mitosis we have the prophase, where during this stage chromosomes condense and become visible, nuclear envelope, for instance, breaks down, and centromeres move to the opposite side. So that's the prophase. The next stage after prophase is metaphase, and during this stage chromosomes line up at the metaphase plate at the center of the cell, and then each sister chromatid attached to the spindle fiber that are at the end of the cell. So here the chromosomes come in the middle of the cell and spindle fibers are attached to them. Then the next phase is anaphase, during which phase the sister chromatids are pulled to the opposite side. So here the sister chromatids are pulled to the opposite side. And then finally the last phase is telophase, okay, during which time the chromosomes now arrived at the opposite ends, and now so therefore they can decondense, and the nuclear material start to form around all these chromatids. So here nuclear material start to form around these sister chromatids. Okay, so that was the function of the centromere in order to allow the spindles to attach to it. And then we have the telomere. Telomere, these are the repetitive nucleotide sequences that are at the end of the chromatid. And basically their job is to protect the actual sequence of the DNA. So here, we have all the euchromatin here, and then telomeres are at the end. So they're providing kind of a protection mechanism by being at the end of the chromosome, because with every DNA replication, the end of the DNA upon removal of the template we just discussed is being shortened. So by having non-coding telomere at the end, it kind of protects the DNA coding sequence. But again, we all know after too many replications, still it can damage DNA and halt replications. All right. The next concept is control of gene expression. And so we know that in an organism, all cells contain the same DNA, but it's a different expression pattern of the DNA that determines their function. And we've discussed in our embryology video that this is called cellular differentiation, where different cells specialize into expressing certain genes, and by doing this they would differentiate into different type of cells. Like it can differentiate into nerve cells, it can differentiate into liver cells, or it can differentiate into pancreas cells. And so by controlling gene expression, cells can have different function, as a consequence of which it's very important to regulate gene expression. Now, gene expression doesn't necessarily need to be controlled at the DNA level. We can basically control it through any steps that goes all the way from DNA to protein, because if we disrupt any one of these processes, then we are interfering with the gene expression. So let me go over some of the mechanisms that can affect gene expression. So gene expression can be regulated at the level of mRNA transcription. We can interfere with gene expression at the level of post-transcriptional modification, or at the level of alternative splicing. So alternative splicing or post-transcriptional modification. It can be done by RNA degradation. So as RNA is trying to reach its target, it can get degraded by different proteins, or it can be halted by certain mechanisms like RNA interference. Then the transport or localization of all the transcriptional machinery could affect the transcriptional process. And then protein translation, as well as post-translational modification. All these processes can affect gene expression. So interruption of any of these processes can affect gene expression. So that's one mechanism of regulating gene expression. Now there are other mechanisms that could inhibit gene expression, and those include, for instance, DNA methylation. So we can inhibit gene expression by DNA methylation. And one example is, like, for instance, lionization in human females. So you know how human females have 2 X chromosomes, but males have only 1 X and 1 Y. So it's too much X chromosomes that are being present in females, and in order not to cause problem, one of these X chromosomes needs to go inactive, and this process is called lionization. And lionization is being done actually by methylation of the X chromosome. So one of the X chromosomes in humans in every cell will become methylated in order not to express gene. And the memory aid that I have for you is that you can always think that DNA methylation mutes DNA. Now we also have another mechanism called histone acetylation. So we can have histone acetylation, which promotes transcription. And how it's being done is that histone acetylation actually loosens the DNA grip, as a consequence of which now it's easier to transcribe those genes. And then we also have regulatory sequences that can affect the rate of transcription. So in eukaryotes, we have a DNA sequence called promoter, and it's actually the site that all the transcription factors, like for instance RNA polymerase, can bind to. So promoter is the site that allows transcription factors to bind to, and this promoter can be affected, its activity can be affected by other DNA sequences, and these DNA sequences include, for instance, enhancers, where these enhancers can have activator proteins that bind to it, and it changes conformation in order to bend the DNA and enhance binding of transcription factors. So by doing this, enhancers can promote transcription process. And then on the contrary, we also have repressor proteins that bind to the DNA coding sequences called silencers in order to inhibit the gene expression, and this is usually done by chromatin modification. Okay, so we just discussed there are different mechanisms of regulating gene expression. We discussed if we interrupt any processes of transcription or translation, that's one mechanism. We discussed that DNA methylation, histone acetylation are other modes of either inhibiting or enhancing DNA transcription. Then we discussed that there are enhancers as well as silencers that can affect the gene expression. So enhancers promote transcriptions, while silencers will modify chromatin structure in order to inhibit transcription. And then the final mechanisms are hormones that affect gene expression. So we can have different modes of activation by hormones. There are some hormones that cannot enter the cell, as an example, like insulin. Once we eat too much food and there's too much sugar in our blood, we need to lower the glucose in our blood, and that is being done with the insulin. So insulin binds the insulin receptors, activates the second messenger, so second messenger molecules, which will affect gene expression. All right, or we could have steroids that directly enter the cell and nucleus and again affect the gene expression. And so basically there are many different mechanisms in eukaryotes where gene expression can be regulated. In prokaryotes, it's not as complicated, but we do have one very famous example, which is the lac operon. And so here I'm going to discuss the lac operon and how it works in prokaryotes. So all the mechanisms of gene regulation that we've been discussing so far was in eukaryotes. Now we've moved on to prokaryotes. And so with this example that was discovered in E. coli, there is a promoter that regulates expression of 3 different genes, LAC-Z, LAC-Y, and LAC-A. So unlike in eukaryotes that every promoter is available for every gene, here you can see that in prokaryotes, a single promoter is affecting expression of multiple genes. So that's one fundamental difference between regulation of gene expression in prokaryotes and eukaryotes. Now the components of this lac operon include, for instance, the promoter, which we just discussed. It's the site where the RNA polymerase can bind to. So promoter, erase that, is the site where RNA polymerase can bind to. Then we have I, or regulator, which is translated into repressor protein. Okay, so here we have repressor gene that can be translated into repressor protein. Next, we have the operator, which is a short DNA sequence that recognizes the repressor protein. So once repressor protein is made, it will recognize operator and bind to it. So let me write that. So operator is the DNA sequence. So DNA sequence that recognizes repressor protein. And then finally, we have the structural genes that are listed right here, like LAC-Z, LAC-Y, and LAC-A. And so Z codes for beta galactosidase, Y codes for lactose transporter, and then A codes for enzymes that actually break down the lactose related products. Now let me show you how does this lac operon system actually works. So under situation where there is low glucose but no lactose, there is not really any need for beta galactosidase enzyme because we don't really have any lactose to use up by the E. coli here. So what happens here is that there is a repressor protein that is being expressed and it binds to the operator. Now you can see that the path of the RNA polymerase is blocked. It cannot go through in order to transcribe LAC-Z, LAC-Y, and LAC-A. And so therefore here, there won't be no expression of the beta galactosidase. On the other hand, if we have low glucose but high lactase, what happens in this situation is that we like to have our beta galactosidase expressed in order to use up lactose for energy production. And so under this situation, lactose products actually bind to the repressor protein, and change its conformation. As a consequence of which, now the repressor has become inactive. It can no longer bind to the operator. And so RNA polymerase is no longer blocked, can go through, transcribe beta galactosidase, which will use up the lactose for its energy production. All right, so in summary, we have two different forms of operons. There are operons that are under positive control. So the regulatory protein that is there is an activator, okay? And once it's on, it will activate the gene. The example that I just gave you here with beta galactosidase is an example of negative control. We have a repressor protein that will prevent expression of genes. And so once the regulatory protein is present, there will not be any expression. On the other hand, if we cause mutation in the regulatory protein, so here, if we cause mutation in the LAC-I, and so there won't be any R protein, then there would be lactase, beta galactosidase expression all the time. So if we cause mutation in the regulatory protein, then the operon on will be on and there will be gene expression. All right, so basically that concludes our discussion of this video. Next, I would like to cover a few topics that I might have not gone extensively in detail. So I'm gonna review that as short question and answer forms, and then following that we will do some practice tests as always, and then conclude this video from there. So the first one was, what is the difference between ribose and deoxyribose sugar units? So we already discussed that. Deoxyribose, which is from DNA, it doesn't have OH on its second carbon, while the ribose have actually an OH group. So remember, ribose was for RNA and deoxyribose was the building sugar structure of DNA. The next question is, what is the difference between nucleoside and nucleotide? Well, nucleoside does not have a phosphate group, while nucleotide has a phosphate group. So the way I memorize it is that nucleoside does not have a phosphate by its side. Okay? Next question is, what is a ribozyme? Basically, these are RNA molecules that have enzymatic activity. And one example of ribozyme, for instance, is a spliceosome, which is involved in the splicing process. What is the relation of the small nuclear ribonucleoprotein particles and small nuclear RNAs? So basically, SNRP, these are splicing factors, that consists of the snRNA that is complex with a set of proteins. So I didn't show it in this image to you, I didn't stress much, but here we are having the snRNPs. Okay, so these are the splicing factors. So again, these are splicing factors that are made of small nuclear RNAs, snRNAs, that are complex with specific set of proteins. The next question is, what is TATA box? So basically, the promoter region of many different organisms have a common feature, and that is these regions are very rich in A and T nucleotides that are located about 30 base pairs upstream of the transcription site, and that is referred to as TATA or TATA box. So this promoter that I was discussing with you is very rich in TATA sequences. The next question is, what is the mechanism of genomic imprinting? Okay, so basically genomic imprinting is that it makes either certain maternal or paternal genes inactive. That is referred to as genomic imprinting, and that is being done by DNA methylation. So for instance, if our body decides to methylate our father's DNA that we've inherited, then the gene from our father would be suppressed. So again, once again, genomic imprinting is DNA methylation that will make certain maternal or paternal genes inactive. The next question is, how do non-coding RNAs regulate gene expression? Okay, so basically we briefly touched upon this topic. There is only 2% of genomic transcripts that code for protein, and the rest of our DNA is basically non-coding DNA. And so these non-coding DNA, for instance, can form RNA interference or small interfering RNAs, which will form double-stranded RNA with our mRNA and then interrupt its expression. So here, assume that we have our DNA transcribed into mRNA. The strand is trying to reach its destination, but then there are some small interfering RNAs that will bind to it and initiate its degradation, right? And so that's how our non-coding RNAs can regulate gene expression. The next question is, what is the significance of gene duplication in process of evolution? Well, if you think of it, if we only have one copy of a gene, it can't really undergo mutation, because if it does and we don't have that gene anymore, what's going to happen to us? We are going to be in disadvantage and we won't really have a good chance of survival. However, if our genes are duplicated, now we have a second copy, and that copy now can undergo mutation, make changes, and come up with a new function that could be helpful. So that's basically the whole purpose of gene duplication, okay? Because that can now undergo changes and come up with new functions. How is lifespan of prokaryotic mRNA different than eukaryotic mRNA? Well, in prokaryotic mRNA, its lifespan is only about a few minutes, while in eukaryotic, it can be as long as a few hours. And can you tell me why is it that prokaryotic mRNA have a shorter lifespan than eukaryotic mRNA? And the reason for that is that prokaryotic mRNA does not undergo post-transcriptional modification. So splicing, 5' cap and 3' adenylation, all those processes are specific to eukaryotic mRNA. And we just discussed how poly-A adenylation will make our DNA molecule more stable, and it's more resistant to degradation by nuclease enzyme. So that's why our eukaryotic mRNA has a longer lifespan, because of polyadenylation of its 3' end. And then the final question is, how are defective proteins tagged for degradation? Basically, proteins that our body doesn't need them anymore are being tagged with the ubiquitin protein, and these are being now recognized by the proteasome enzymes that will break down those proteins. All right, so we are basically done now covering all the short questions as well. So here is some practice questions to go through. So I'm going to read these questions for you. If you need more time, by all means, you can pause the video. Which of the following is true regarding Okazaki fragments? Which of the following enzyme unwinds DNA helix? Well, after going through these lectures, all these questions sound so easy. The beginning of genes that code for protein is at, well, this is a little more challenging question that needs you to think a little about it. So basically it's asking your mRNA. You got to think of your mRNA. And then from there on, you can predict where did it come from. Did it come from template, from coding DNA strand, and is it corresponding to the 5' or 3' end? Okay. And I highly urge you try to solve this question, because MCAT really likes to ask these questions and try to confuse you with how the 5' end of one DNA correspond to the 3' end. All right, next question. Which of the following forms of DNA repair takes place during replication? Okay. Next question. So it's during replication, active replication. Which of the following forms of DNA repair is dependent on helicase enzymes? And then let's stop right here, answer these questions, and we will come back. So let's go back to the top and answer all these questions. First question. So again, if you need more time, please pause the video. Otherwise, we are ready to go through. So which of the following is true regarding Okazaki fragments? So if you recall, Okazaki fragments were the fragmented DNA sequences that are shown right here. Okay? These are the Okazaki fragments. They are on the lagging strands. So they are found on the lagging strands. So both choice B and D so far are correct, and they're synthesized in the, so doesn't matter what strand we're talking about or Okazaki, synthesis of RNA and DNA is always in 5' to 3' direction. So therefore, they're synthesized in a 5' to 3' direction. So again, I don't want you to be confused. Both the leading and lagging strands and mRNA transcription, everything is in 5' to 3' direction, meaning that the new nucleotides can only be added at the 3' end. All right, the next question is, which of the following enzymes unwinds DNA helix? Again, that's very easy, helicase. What was the function of topoisomerase? It forms nicks in the DNA in order to relieve the supercoils that are created by helicase. So does gyrase. Both of these does the same job. And ligase, what was the function of ligase? It was joining the Okazaki fragments together. Now this is my favorite question. The beginning of a gene that codes for protein is at? So here is how we summarized our question. You can see that the beginning of our RNA, the 5', corresponds to the 3' end of the template DNA strand. So therefore, our very first protein corresponds to the 3' end of a template DNA strand. So choice 3 would be the correct answer. 5' end would be incorrect because that would be our last amino acids. Next question. Which of the following forms of DNA repair takes place during active replication? So here I was referring to proofreading as we are incorporating new base pair. If there is a mistake, then it would remove that by the DNA polymerase. But then mismatch repair, as we are going through DNA replication can also go through and make modifications. So if you have chosen either choice A or choice B, I would give you credit for it. I should have been more specific with this question. All right, and then the next question is, which of the following forms of DNA repair is dependent on the helicase enzyme? So which one was the most simple one? It was base excision repair. It didn't need to remove any segment of DNA. Versus nucleotide excision repair and mismatch repair, both of these had to remove a large segment of DNA. So both of these, number 1 and number 3, are dependent on the helicase enzyme. Let me go back right here. So here we had base excision repair. We didn't use any helicase because the only defective base, we've just removed that one. Versus with nucleotide excision repair or mismatch repair, we removed a large segment of DNA. And for that, after nicking with the endonuclease enzyme, we used the helicase to remove. So now we are done with the first set of questions. Let's move on to the last set. Which of the following DNA mutations always shortens the size of a translated protein? The next question is, which of the following is the first step to initiate gene expression? Then we have, which of the following structures are transcriptionally inactive? And then the final question is, which of the following is a true statement regarding the control of an operant system? So we are talking about prokaryotic systems. Okay, so if you need more time, please go ahead and pause the video. Otherwise, we are ready to start. So which of the following always shortens the size of translated protein? Well, we discussed stop the nonsense. It's the nonsense mutation that will convert a sequence into a stop codon, as a consequence of which there could be truncation. Deletion or addition or frame shift mutations, they could potentially truncate a protein depending on how it switches the amino acids, or it could make it longer because we cannot predict the whole DNA sequences will be, I should have said the whole triplet codons will be changed by either deletion or addition because they cause frame shift. And then we just discussed missense is only changing a single amino acid, and so it doesn't necessarily truncate a protein. Next question is, which of the following is the first step to initiate gene expression? So in order to express a gene, the first thing that we need to do is to unwrap the chromatin in order to make it available for gene expression. And that's why histone acetylation, so acetylation of histone, it helps with relaxing the DNA. So that's actually one of the very first steps before we can initiate gene expression. The next question is, which of the following structures are transcriptionally inactive? So methylated DNA, yes, that's a correct answer. Heterochromatin, we discussed, it's very condensed, tightly packed, so that is also transcriptionally inactive. Acetylated histone, we discussed, that is actually activating transcription. So acetylated histone activates transcription, while methylated DNA mutes DNA. Okay, so choice 1 and 2 would be the correct answer. And then the final question to answer right here is, which of the following is a true statement regarding an operant system? Mutations in activator turns off positive control. Okay, so activator, we just discussed that our positive control has activator while negative control has repressor. So here, activator has positive control while repressor is a mechanism of negative control. If we destroy activator, the gene will no longer get expressed. On the other hand, if we destroy repressor, now we can express the gene. So now, with that review, let's go back to the question again. Mutations in activator turns off positive control. So that is correct. Mutations in activator causes constitutive expression of genes. Actually, if we don't have the activator expressed and there is mutation in that, there won't be any expression. So that would be incorrect choice. Next, we have activator is the regulatory protein in the negative control system. No, that's in a positive control system. It was a repressor that was in a negative control system. And then finally, mutations in repressor renders an operon system off. So what happens with the mutations in a repressor is that it doesn't turn off the operon anymore. So it should be that it renders an operon system on. And so here, let me show you right here. So if you have a mutation in a repressor, it renders an operon system on, as a consequence of which now the gene can be expressed. All right, so that was with the last question, which makes choice A the correct answer. And so that was it for our lecture on transmission of genetic material from gene to proteins. I hope you found this lecture helpful. Thank you all for listening, and we will see you for the next lecture. Thank you.